In this video, we're going to discuss the sequence of events in the immediate type 1 hypersensitivity reactions. We're going to talk about kind of in detail what goes on during this type 1 reaction. And this is an interesting video, and I've tried to put a lot of detail in here, and I've loved learning about it because I have allergies, seasonal allergies, and so it's interesting to learn about what's going on with you. Anyways, so the first thing that happens is you get exposure. You get exposure to the allergen, the pollen, the bee venom, whatever you're allergic to, food allergies, what have you. And the allergens are introduced by inhalation, ingestion, and injection. And the allergy response has to do with if the innate immunity or the natural killer cells or whatever, if they're here and they just gobble up this allergen real quick, if the innate immunity kind of rids itself of this allergen, well then the B cells, because this is really the adaptive immune system, you don't really get sensitized. And that's the first thing, is that you have to have exposure to the allergen, you have to become sensitized, you have to be exposed to it first, and then the second time is when you start noticing symptoms, allergy symptoms. So if the innate immunity kind of catches this, then you're not gonna have the allergy. Also, the genetic predisposition of the individual, whether how long and how chronic the allergy, what kind of dosage, that how many allergens of the same type you were exposed to, all that kind of plays into kind of the allergy response or how severe your allergies are. But the means of introduction is the inhalation, ingestion, and or injection, if you will. So what happens is you get exposed, and then the allergen goes through the mucosal layer and then attaches, the B cell picks it up. Now what happens is when the B cell picks it up, it sends out chemokines. Okay, and the chemokines recruit this cell right here, which is the T helper cell version 2, if you will. And it also, another chemokine is the eotaxin. And the eotaxin, the substance right here, it recruits eosinophils. And we're going to talk about how the eosinophils play a role in this kind of hypersensitivity reactions. So the eosinophils are recruited, and also the T helper cells are recruited. So once the T helper cells they kind of make a connection here, and the B cell kind of communicates with its T helper cell. The T helper cell secretes cytokines, okay? And the cytokines that it secretes is the interleukin 4, which stimulates the B cells to produce IgE antibodies. Because this could be like, there's a little, if you don't know all the different types of antibodies, there's a kind of a little word, if you remember, gamed. You got played, you got gamed, if you will, but GAMED, those are all the IgG, and then you just substitute GAMED into the IgG. Those are all the antibodies. But this could be like an let me change colors here. This could be like an IgM antibody right here. Okay, so instead of secreting IgMs, because this is the one that picked it up, this interleukin 4 stimulates these B cells to kind of class switch, if you will. Right here, they kind of class switch, so it switches to a different class, the IgE, and so then you get an increase in the IgE antibody production. Okay, now that's interleukin 4. That is the cytokine that's secreted from the helper T cell. Then another cytokine that is secreted from the helper T cell here is this interleukin 5. The interleukin 5 activates these eosinophils that were recruited by this eotaxin from the chemokines that were secreted from this B cell here. Okay? And then the next thing is the interleukin 13, which acts on epithelial cells and it stimulates mucus secretion. So you start getting mucus secretions. You start getting in your nose, rhinitis, runny nose, if you will. So those are the cytokines that are secreted from the helper T cell. So now once you have this increase of IgE antibodies, these IgEs, they kind of come down here and they sit. They sit like this in this receptor. This receptor is mainly on mast cells here. Now, this is the IgE antibody here, and this is the IgE receptor. Now, mast cells right here, mast cells express a high affinity receptor for the Fc portion of the ypsilon. This is an ypsilon, the fifth letter of the Greek alphabet, the ypsilon heavy chain of the IgE called the Fc ypsilon Ri. What that means is this mast cell has a receptor. Okay, and this receptor has a very, 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 very high affinity or a high like or love of this Fc portion of the ypsilon heavy chain of the IgE antibody. Okay, so if you don't know what that is, let me kind of scroll down here. I got this picture from Wikipedia. So right here, so this is an antibody here, and the antibody is broken down into two parts. Number one is the FAB region, which is the top half of the picture here, and then FC region is the bottom half. So they're talking about this FC region here. And the heavy chain is this big kind of blue, these shades of blue. This is the heavy chain of the IgE or the antibody. And then this is the light chain. So this heavy chain, this FAC region, this lower portion here, this receptor has a high affinity for that receptor. So even though the concentration in your blood is very low for IgE, pretty much all the sites on mast cells, there's IgE sitting there because there's such a high molecular, if you look into the biochemistry of this, there's such a high affinity for those molecules to be by each other. And I forgot to mention that mast cells, they are produced in the bone marrow and they live all over your body. Okay, they're all over, and they're camp out mainly by blood vessels, by nerves, and subepithelial locations, kind of strategically located that they can intercept, these IgEs that are floating in the blood. They could be a part of the immune system here. So mast cells, basophils, and eosinophils have a high affinity for this Fc ypsilon Ri. So they have a high affinity for these IgE antibodies. Okay. Now, remember how I talked about up here that these eosinophils are recruited? The eosinophils also play a role, immediate hypersensitivity disease, but eosinophils are mainly responsible for helminth infections. And if you don't know what a helminth is, this is a parasite. So when you have a parasitic infection, this is why these eosinophils are recruited. So you're probably thinking, well, why do we even have this pathway? Like, why does our bodies even have this pathway? Is it because so we can have allergies? The answer is obviously no. We have this pathway mostly for parasitic infections and other infections, but our body responds, obviously has an erroneous pathway or utilizes this pathway erroneously when it comes to allergies. So mainly this pathway is for parasitic infections and different things that we'll talk about later in the video, but this system or this pathway is used in the wrong way, and that's how we develop allergies. But these mast cells and basophils are circulating in the blood. They're kind of the analogous to the mast cell. The mast cell is outside the blood, the basophil is inside the cell, but they're pretty much the same cell type. But these basophils are in the blood. Okay, so now that you have these IgE antibodies that are attaching to this IgE Fc receptor on the surface of these mast cells, you need two of them kind of together with an antigen binding to both of them, and what that does is that activates. You have the activation. So right here, this is kind of the activation step. Activation step. Where then once this is kind of this situation is encountered, this situation occurs, then you have some few things that happen. The first thing is this degranulation. So if I put a 1 here, we have, first thing we're going to talk about is this degranulation step. These degranulation step means that there's prefabricated inside these little pouches some goodies. And these mast cells, when they're triggered by this activation step, they degranulate. And what is in these granules, or these little goodies? You have histamine, proteases, and chemotactic factors. So here there's kind of a laundry list of what's in these kind of granule contents. Number 1 is the vasoactive amines. Okay, so the first part is vasoactive amines. And what's the number 1 thing is histamine. Histamine is released from these mast cells. And what does histamine do? It causes vasodilation and increased vascular permeability. So if we have here a normal blood vessel and it is acted upon by histamine, what happens is the vessel becomes bigger, and then there's little holes. There's little holes that are formed, and that's vascular permeability, so that the blood and white blood cells and different things can kind of leak out. Vasodilation, increased vascular permeability. Histamine also causes smooth muscle contraction, and it increases the secretion of mucus. Another thing is this adenosine. Adenosine causes bronchoconstriction, and it inhibits platelet aggregation. So you have in your lungs, you have constriction, bronchospasm, you have constriction of your lung, the airways in your lungs, so you can't breathe as good, and you have inhibition of the platelets. So your blood clotting doesn't happen as good as it should. Another important thing is you have chemotactic factors for neutrophils and eosinophils. And if you don't remember what chemotactic factors are, I'll explain it. Okay, so you have these degranulation, and so if you have a war here, in this case it's a war between this antigen or this allergen against this mast cell. Your body doesn't know that this thing is harmless, so it takes the necessary steps to recruit all your white blood cells, most of them, to this war so that they can help fight the war. Say this is a neutrophil here. This cell has a little N in it, so it's the neutrophil. And as this concentration, or as these chemotactic factors are released here, there's going to start to fuse into the tissue. They're going to start to fuse, and with time, you see these little dots all over. That's diffusion of these chemotactic factors. And these neutrophils and eosinophils, they can kind of sense the concentration of these chemotactic factors, and they will migrate to the greatest concentration of these chemotactic factors, and then they will, then they'll be here and they're like, oh, hey, this is the problem, and then they'll start fighting. But in the case of this hypersensitivity immediate type 1 reaction, or disease, if you will, it's an allergen. It's not harmful. But nonetheless, this process happens, which is part of the inflammation response. Next, you have these neutroproteases, and this kind of goes back to these eosinophils here. Is this process normally happens when we have parasitic infections or other type of infections, as helminth infections. And these neutroproteases what they do is they also damage tissues because they'll go in there and they're proteases, so they'll chew up proteins. So they're going to chew up proteins, which is going to damage actually tissues. They're going to generate kinins. They're going to cleave complement components, which help with the chemotactic and the inflammation part. But another thing that they do that's really important is since they're proteases, and let's say, for example, you get bit by a snake, some of that venom is proteins. So these proteases will go and they'll deactivate that venom, the proteins inside your snake venom. So that's an important part of, that example, is that these neutral proteases will deactivate foreign venom, if you will. And another thing is the acidic proteoglycans and the heparin and chondroitin sulfate. These are just storage matrix for the amines. So they provide a matrix for the amines to kind of be stored in. So that's step one, the degranulation kind of process. The second thing that happens is this membrane's phospholipid. You have the platelet activating factor, you have arachidonic acid cycle, and the arachidonic acid is broken down into prostaglandins and leukotrienes. So part 2 here is you have newly synthesized lipid mediators. You have prostaglandins and leukotrienes. So prostaglandin D2, PGD2 is kind of the short version of it. It undergoes the cyclooxygenase pathway and goes down and follows that, and I've done videos on that in the past and you can watch those. There's an intense bronchospasm and you have increased mucus secretion. That's what prostaglandin D2 does. Leukotrienes C4, D4, they are the most potent vasoactive and spasmogenic agents known. They're very powerful. Histamine, as we talked about up here, does vasodilation and increased vascular permeability, while leukotrienes is like 1,000 or 2,000 times stronger than histamine. They're very powerful. So you get a teaspoon of this in your system and you're gonna be in trouble. So that's why these membrane phospholipids are kind of released to kind of follow up with the job of histamine. Leukotriene B4 is highly chemotactic for neutrophils, eosinophils, and monocytes. So we see the recruitment of more white blood cells. And it's redundant. There's a lot of redundancies, duplicates, if you will, in kind of what happens over here and what happens over here, is because, if part of your system doesn't work, well, then we still get the job done. And the vasoactive amines that are released from these kind of storage sites, if you will, and these membrane phospholipids, they are responsible for this immediate response. And the immediate response is vasodilation, vascular leakage, and smooth muscle spasm, which is, responsible for bronchospasm and things like that. And then the last thing that's secreted by these mast cells after this activation is, you get some signals from some cytokines that cause gene activation. So you're inside your DNA, inside your nucleus, the DNA is read, translated, transcribed, things like that. And then proteins are produced in the Golgi, outside in the cytoplasm, and then they're shipped to the Golgi for packaging and shipping. They get shipped out, and then you get this late phase reaction. I'll explain to what late phase reaction means in a moment, but you can kind of get a hint of it, that it's late. It comes on later. So what you get in this late phase reaction is you get leukocyte infiltration, epithelial damage, and more bronchospasm. Epithelial damage is because these leukocytes, they're not really nice. They come in and they get the job done, and in the process, they destroy some of the tissue. In the case of this type 1 immediate hypersensitivity reaction, they're destroying tissue for no reason, because the simple allergen right here is not harmful to you, but their body thinks it is, and so you get this type of reaction. So what happens is you get cytokines, and this is, let me just so we're clear here, this is the kind of the third step that happens. You get cytokines, tumor necrosis factor, and chemokines. They recruit and activate leukocytes, so more activation, more recruitment of the white blood cells. Interleukin 4, interleukin 5, they amplify the T helper cell 2 initiate immune reaction. So it's kind of a positive feedback loop to this step here, where you get kind of an increase in this process right here where there's cytokines being released by this helper T cell, version 2. And you also get interleukin 13 released, which stimulates more of mucus secretion. So let's go over here to this picture here. And this picture was also taken from Robbins' Basic Pathology 8th edition. What happened is, this is kind of a graph here. This is the signs and symptoms, clinical manifestations on this axis, and then on this axis you have the hours after the allergen exposure. So first you have the allergen exposure right here, and the immediate part, like type 1 and 2 over here, part 1 and 2, you get this immediate response. This kind of happens quickly, and you kind of see this a lot of signs and symptoms, a lot of clinical manifestations, and then after 30 minutes or so it kind of dies down. But then you get this late phase reaction, and so you kind of get another hump in the signs and symptoms. After 4 to 16 hours, you kind of see this late phase reaction. And why is that? Is because you're getting these leukocytes that are coming to the area now, because it takes time to get the leukocytes there. You start seeing a lot of this epithelial damage. Tissues are getting damaged because these leukocytes are going in there and they're wreaking havoc. They're releasing stuff, proteases or other enzymes, and they're chewing up tissue. So you kind of get these, you can get more bronchospasms. So that's kind of what this late phase reaction in this type 1 hypersensitivity immediate reaction is. And in this picture you can kind of see, this is a slide of cells and different things of tissue. You can see this vascular congestion inside this blood vessel. You can kind of see this edema coming out, swelling, tissue swelling, and then you see these mast cells and they're releasing these degranulation type stuff. And then again in the parasitic infections, and mostly for parasitic infections, that's what these eosinophils respond to, but this type 1 scenario reaction also recruits these eosinophils here. So we'll see you in the next video.